Good morning, student. Today I am going to discuss about the first chapter in immunology called immunity. So these are my learning object. So you will end of the class, you will aware of what is it, immunity, how it is classified, and what are the cells are involved in this immunity. And let me give the definition for the immunity. Immunity, it is a resistance offered by the host against the microorganism or any foreign substances. So it is broadly classified into innate immunity and acquired or adaptive immunity. What do you mean by innate immunity? Innate immunity, nothing but that resistance they got at the birth itself. So, but in case of acquired immunity, it is acquired during the course of life. This is the difference between innate as well as acquired immunity. Let us discuss about the difference between acquired and innate immunity. As I already mentioned that innate immunity nothing but the immunity defense mechanism acquired at the birth itself, whereas this acquired or adaptive immunity, they have the resistance offered during the course of life. This immune response act immediately when the foreign antigen enter, the immune cells will encounter the antigen. It occurs immediately within a fraction of second. But in case of in acquired or adaptive immunity, it takes some times. But in case of innate immunity, it needs some kind of antigenic. It's not necessary to have a prior antigenic exposure. So it has a diversity, it will be limited. Only particular antigen it will react and it will offer the resistance, not entire antigens. In that, because of it is a, does not have a prior exposure, so it has less, I mean, it is lack of memory reaction. It won't recognize the antigen when it encounter again. So there is a specific, there is no specific antigen antibody reaction, but it has that antigenic unit called microbe associated molecular patterns. If the antigen contains this, the host cell has a toll-like receptor called pattern recognizing receptor. So when antigen has that molecular associated microbial pattern, it will bind to the toll-like receptor, which has the pattern recognizing receptor, and it react and it kills the bacteria or any kind of foreign antigen. This is the about the innate immunity. When it comes to the acquired or adaptive immunity, the already have mentioned the definition. It occurs during the course of life. So the immune response not occurs immediately. It takes some times to activate the T cell, B cell, and development of that kind of defense. It needs a prior exposure of the antigen. And if the antigen is exposed, it is processed and presented, then only it get activated. So because of this long process, it has some kind of memory, and it is specific to the T cell react to the some kind of antigen, B cell will react some kind of antigen. There is a specificity is there in compared to innate immunity. It has a specificity. So it has the T cell receptor, B cell receptor, whereas in case of innate immunity, it has a toll-like receptor. That toll-like receptor called pattern recognition receptor. Okay, so this is of the innate immunity and acquired immunity differences. So just highlighting the few points about the innate immunity. It occurs during at birth itself. There is no prior exposure of the antigen, no memory cells. There is a diversity will be limited. It has a molecular that associated microbial pattern, and it will bind to the toll-like receptor which is present in the host itself. When coming to the acquired immunity, it occurs during the course of life. The immune response takes some time, and it needs antigen exposure, prior antigenic exposure, and because of that, it has a memory. So that second time, if it is encountered, it is able to recognize the and defend immediately. So it has a T cell receptor and B cell receptor. Coming to the cells which is involved in the innate as well as acquired immunity, There are few cells which is participate in the innate immunity called that anatomical structure, anatomical barrier called skin and mucosa. It is normally present in our body. And phagocytic cell, natural killer cells, macrophage, dendritic cells, and some kind of complement like alternate and lectin pathway, and some cytokines which is produced as a secretory molecules. These are the things will come and play in the innate immunity immediately. When coming to the acquired immunity, it has a T cell, B cell, classical pathway of the complement, and some kind of cytokines. So this classification will be discussed later. We'll move on to the mechanism of the innate immunity. So the innate immunity, it has first, I have already mentioned that it should recognize and bind to the host cell receptor. So the organism, that is microorganism, has that molecular associated microbial pattern that will recognize the toll-like receptor. This can be present either in the macrophage dendritic cells. So once it is recognized, it will go and attach to this kind of cells called pattern recognizing cells, so that it is in once attached, it is engulfed, and if suppose phagocytes has this kind of pattern, so this toll-like receptor taken up that microbes and engulf the phagosomes will form the and lysosome which is present in the phagocyte that fuse and form the phagolysosomes. Then later the cells are killed. This is called receptor interaction. Uh, this already have mentioned that toll-like receptor will be present in the host cell. This microbes associated molecular pattern will be present in case of any kind of microbes. For example, if the bacteria has the peptidoglycan, lipopolysaccharide, teichoic acids, lipoprotein. These are the parts act as a microbes associated molecular patterns. This toll-like receptor which is present in the host, like pattern recognizing receptor which is present in the host, these two will interact and kills the antigens. So, which I already mentioned that few cells are involved in the innate immunity, like these are the few cells which is responsible for the killing of the bacteria via the innate immunity, like anatomical and physiological barrier, and phagocytic cells, natural killer cells, mast cells, dendritic cells, some class alternate and lectin pathway, and inflammatory response which is produces secretory cytokines and some kind of acute reactant proteins. So these are the cells which helps to activate and destroy the microorganisms. Let's discuss the one by one. Now we'll discuss about the anatomical physiological barriers. Like example, skin, it is present in the birth itself. It is the largest organ. So that sebum is there in the skin. It acts as an antimicrobial property that kills the microbes by intraepithelial lymphocytes. This intraepithelial lymphocytes which is present in the skin that engulf and kills the microorganism. Coming to the other physiological barrier called mucous membrane, which kills the bacteria or any microorganism by entrapment of the microbes. Cilia, which is present in the respiratory tract, that propel the microbes outside. So instead of getting inside, it is propel outside so that the microbes will come out and kills. And normal flora, this is the main intestine, respiratory tract, wherever the skin, they have the normal flora which competed with the, it has a competition between the pathogenic flora and normal flora. So normal flora has the more capacity to kill the bacterial or any kind of things if it is less number. So it has a competent competitions. And coming to the temperature, normal temperature inhibit the growth of the some bacteria or viruses. And if the pH is low, that is acidic environment, the bacteria fail to survive in this condition. So it makes the environment free of the microbes. and some kind of secretory product which is secreted in the mucosa like saliva. So saliva has the enzyme, which always enzyme breaks down any kind of things. So the bacteria cell wall, cell membrane, it is breaking down and inhibit or stop the growth of further microbes. And tears has the lysoenzyme that also helps to destroy the bacterial cell wall. Gastric juice, gastric acid, HCl, it is also helps to because of the acidic nature, there is a less chance of bacteria to survive in that environment. And like this, these are the few points favoring that innate immunity occurs at birth itself. It's the anatomical, it normally present at birth. So like this, we are not getting any kind of infection because of our proper innate immunity during life. Little bit about the phagocytes. Already have mentioned that any bacteria, any foreign antibody, foreign antigen, it has a microbes associated molecules pattern. The phagocytic dendritic cells has a toll-like receptor. It binds and the bacteria enter inside, called a receptor interaction. Once enter, it is called a phagosomes. These phagosomes fuse with the phagolysozymes. Okay. Once again. So this phagosomes which fuse with the phagolysosomes and called phagosomes called a phagolysozyme. Okay. So one it is enter inside this lysosome because of the enzymatic action, it kills the bacteria so that it kills the bacteria into fragment and it come out. Likewise, the phagocytes kills the bacteria. Okay, first engulfment and phagolysosome microbial killing. These are the three steps which is involved in the phagocytic mechanism. Coming to the natural killer cells. Even though it is a part of the innate immune cells, it more active in case of adaptive or like later kind of in adaptive immunity only. This is even though it is a part of the innate immune cells, it's more supporting case of adaptive immunity. And other classes of the lymphocytes which also involved in the innate immunity, like usually alpha beta cells are present in the T cell receptor. In rare cases, this gamma delta cells also plays in innate immunity to kill the microorganism. NKT cells, B1 cells, marginal zones of B cells. These are the cells, even though it is rare, it is helps in the killing of the microorganisms. Coming to the mast cells. Mast cell usually present in the mucosa or in the respiratory tract. It is activated by the microbial product called that is microbial associated antigen. So it is present in the antigen, and the mast cell has that two things. One is toll-like receptor or IgG antibody. So this toll-like receptor or IgG antibody will bind to the antigen so that it kills the with the help of enzyme which is present in the mast cell. So the mast cell has this enzyme. So this is called IgE antibody, or sometimes toll-like receptor will be present. Suppose there is a IgG antibody coated mast cell, it is there. If there is any kind of antigen comes, it react and engulfed, so that with the help of enzyme, the granules are produced. With that granules, the bacteria are destroyed. So this is the mechanism of mast cells. Coming to the dendritic cells, it is a part of the innate immune cells, but it is participating in the acquired immunity. It makes a bridge between the innate as well as acquired immunity. Once the antigen enter in our body, it get attached to that and it transport to the lymph. It activate the B cell or T cells. Depending upon the antigen nature, the T cell or B cell get activated. Coming to the complement pathway, I already mentioned that innate immunity, like alternate or lectin pathway helps in removing the microbes in case of innate immunity. Whereas adaptive immunity, classical pathway plays a major role to eliminate the microbes. So here already mentioned alternate and manners binding pathway are the mediated by of the innate immunity. So it activated in response to bacterial endotoxins. If the bacteria has the produce endotoxin, always gram-negative bacteria produce the endotoxin that get activate the alternate pathway so that microbes were killed by in that way. Coming to the inflammatory response, when antigen antibody are interaction, there is a biological response of the tissue, inflammatory tissue, leads to the production of the inflammatory response sign. Okay. So what are the inflammatory signs can be observed? Okay, imagine this is the any tissue damage. Okay, tissue damage happened here. Once tissue damage had happened, leads to vasodilatation. That is because of the capillary permeability. Okay, once the capillary permeabilities are happened, vasodilatations are and leakage of the fluid. So it start migrating all the inflammatory cells and extravasation of the phagocytic or inflammatory cells or any kind of innate immune cells. Once it is extravasated, this kind of phagocytes are engulfed. Once it is engulfed, then the fusion of the microbes as well as the phagosomes. These phagosomes are fused with the lysosome and called a phagolysosome, then killing of the bacteria, which already discussed earlier. So I already mentioned first initially there is a vascular permeability, migration, rolling of the endothelium, extravasation, chemotactic migration of to the inflammatory site. This already repeated only engulfment of the bacteria, destruction of the microbes. Okay. What is the role in normal resident flora, normal flora? So normal flora has a competition between the pathogenic flora and it resists to set the pathogenic flora in the normal site. So it has a compete with the pathogenic for the nutrition. In order to survive in the, for example, skin, there is a Staphylococcus epidermis which is present normally in the skin, which is a normal flora. So in order to survive that Staphylococcus epidermis, it will not allow any bacteria to settle in that. That is the competition between the pathogens for the nutrition. And it will produce the antibacterial substance. For example, in the intestine, so many gram-negative bacteria will be able to produce a biocin. Biocin, nothing but it is an antibacterial substance which kills the other than that species, other than that strain, it is able to kill the other. So that other bacteria is unable to survive in that environment. Okay. Coming to the cytokines, in response to the microbial antigens, some dendritic cell macrophages produce several cytokines that mediate many of the cellular reaction of the innate immunity. For example, it is able to produce a tumor necrosing factor, interleukins, interferon, and transforming growth factor. So because of these things, the bacteria are able to kill. Coming to the acute phase reactant, which is one of the short notes for you. Okay. One of the short notes. So the protein which is synthesized by the liver and other various like endothelium, fibroblast also, mainly it is synthesized by the liver only. In two extent, either it can be increased or it can be decreased. If the inflammation is there, either the inflammatory that acute phase reactant can be increased or decreased based on the condition. You have to think about based on that increasement or decrease. It is called a positive acute phase reactants or negative phase reactant. Acute phase reactant means there is an increased above the normal range, that is called a positive acute phase reactant. So let's take the example: C-reactive protein, complement protein, coagulation protein, proteinase inhibitor, alpha-1 acid glycoprotein, metal binding protein. These are the proteins, or these are the factors will be increased during the inflammation. This is called a positive acute phase reactants. Coming to the negative acute phase reactants, the proteins whose levels are decreased during the acute inflammation called it, because of some kind of the inflammation, it levels goes beyond the normal range. Example, albumin, transferrin, antithrombin. In case of any coagulatory disorder, these things will be decreased. This is called a negative acute phase reactants. So what is the role? Why we are checking whether increased or decreased? Why? What is the purpose? The various antimicrobial, anti-inflammatory activities are there. So metal, for example, metal binding protein can chelate various metals such as iron, copper, etc., making them unavailable for the bacteria. That is why we are checking this. Okay. So if there is any change in this pattern, we can think about some abnormalities there. I'm going to discuss about the CRP. Maybe CRP is one of the short answer. So I'm going to focus on that. Already told you it is a positive acute phase reactants. That means the level goes up. So one thing I already mentioned that it is synthesized by the liver. One thing. Second point, it is a beta globin family. And the CRP is named why the C reactive protein. It able to precipitate with the C carbohydrate antigen of the pneumococcus. There is a once things will be present in the pneumococcus. Pneumococcus is a bacteria, gram-positive bacteria. The cell wall contains C carbohydrate antigen. So what happened? This C reactive protein are able to react with the C carbohydrate antigen of the pneumococcus. That's why it is named C reactive protein. But it is not the antibody against the C carbohydrate antigen of the pneumococcus. Okay, that you should remember. It is not the antibody against the pneumococcus. And then it is raised any inflammatory condition, not only the pneumococcal infection, any inflammatory condition like knee joint infection, inflammation, heart problem, any inflammation occur in body, the CRP level goes up. It is an inflammatory marker. That's why it is a positive acute phase reactant. And coming to the other, what is the purpose? When it level goes beyond 0.2 milligram per deciliter, it is called abnormal. Why there is a cutoff value? Because it's already, I told you, it is synthesized by the liver. That is the normally some amount will be present, right? That is 0.2 milligram. If goes more than 0.2, okay, that is the range you can able to tell this is the abnormal. So they have a cutoff value that 0.6. 0.6 is a cutoff value. When it goes to more than 0.6, it indicates that is positive. That is CRP reactive. So we can, based on the title range, we can classify significant means more than 10. It is a markedly significant. It happens in the acute bacterial infection or any major trauma, any vasculitis. In this condition, the CRP value goes up. It goes more than 10. The moderately increase called 1 to 100, 10, sorry, 1 to 10. In some case of bronchitis, respiratory problems, cystitis, malignancy, pancreatitis, myocarditis, in this condition, the cut value will be 1 to 10 only. Insignificant, when you have to tell that is not significant at all, less than 1. That's why it already 0.6 milligram per deciliter. It can happen even if you done heavy exercise or any common cold, or in case of pregnant woman, the level will go 0.6 to 1. So you have to take a counter. If goes more than 0.6, that is accountable as a significant value. Okay, how can we able to detect the CRP value? That is precipitation method. Was done in the previously. Nowadays it is not done because we have latest method to detect the CRP value called latex agglutination. that is using the latex particle. It is available commercially. If you use that kit, you can able to tell whether the positive, negative. There is a agglutination. There is a visible clumps will be seen because of the latex particle. So based on that, you can tell this is the positive. It is more than 0.6. It's less than 0.6. Based on the observation by the naked eye itself, you can tell. That is called a latex agglutination test. What's highly sensitive? You have done this method. It is sensitive. Like nephrometry, enzyme immunoassay. If some ranges come, you should not ignore it. It is a gold standard method. You can go for the treatment modality. So this is how we can able to diagnose the CRP. I'm summarizing the CRP. CRP nothing but it is synthesized by the liver. It is a beta globulin family. It react to the C carbohydrate antigen of the pneumococcus, but it is not the antibody to the pneumococcus. So how we can able to detect? We can do done by the latex agglutination method. The latex agglutination method, we can be able to see the clumps. So if it does go more than 0.6, then the clumps will be visible. So there is a gold standard method called nephrometry and enzyme immunoassay, by which you can able to tell. And based on the titer value, there is a moderate increase. If it is more than 10, you can say it is a it is moderately sensitive. And some insignificant like heavy exercise, pregnant woman. So we can have a little higher value. Now we'll move on to the acquired or adaptive immunity. Let me discuss or summarize the innate immunity in short. So first thing, innate immunity occurs during at birth itself. First point, it is not having any memory cells. It is not needed for the prior exposure of the antigen. The diversity of the things will be limited, and it has a microbe associated molecular pattern which go bind to the toll-like receptor which is present in the host cells. And there are several cells are involved in the innate immunity, like example, anatomical barrier, physiological barrier. Anatomical barrier, we have the skin, it has a sebum. And the physiological barrier called a mucus, okay, cilia. These are the things which helps to kill the microbes. And few cells like cytokines, inflammatory cells, acute reactive proteins, and these are the things which helps in the innate immunity. Okay, so now we'll move on to the acquired or adaptive immunity. This is another important note that you have to understand. Okay, acquired or adaptive immunity, nothing but already I mentioned that it occurs an immunity defense against the microbes, but the individual acquire or adapt over a period of life during the adult period when birth happened during the period of life. Okay, it is again divided into active immunity, passive immunity. Active and passive immunity, further it is divided into natural artificial and natural artificial. Okay, coming to the active natural, if there is any infection, like example chickenpox. So once infection occurs, the immunity will be lifelong. We have acquired the immunity by actively and but infection naturally. Naturally, we have got the infection and the immunity developed throughout the lifetime. And coming to the active artificial, there is a vaccine. Okay, how we are getting immunity by the vaccine? That is comes under active, but artificially we are getting. Coming to the passive immunity, then naturally the mother to fetus, naturally it is going, but in a passive manner. Likewise, passive artificial, there's some kind of and immunoglobulins. So monoclonal antibodies, these are comes under passive artificial. Okay. So most important things we have to remember this classification. If you see this picture, then only you can able to understand you can able to understand the things. Okay, and like acquired immunity, we already have mentioned that it is acquired during the life period, and it has a memory, and it is as a diverse group. There is an antigen specificity. B cell react to the specific antigen, T cell react with the specific antigen, and some it needs some kind of prior exposure that I already mentioned. Okay, and it needs a memory. Okay, the T cell B cell will recognize if same antigen will comes again, it will able to recognize the that's why it is called memory will be present. And diversity is wide. Acquired immunity though takes time to develop, it takes some days to develop the immunity, but it is a wide range. Okay, it is a wide range. And host and receptors. Already I mentioned T cell, has a receptor. The antigen will bind to this kind of receptor, and the reaction takes place. So this picture already I have mentioned. Active immunity following in exposure to the microbial infections. Artificial means develop due exposure to the immunology by the vaccines. Passive natural means involves the transfer of the antibody, and that is transfer from mother to fetus. Usually IgG cross the placenta, and the babies get the immunity. Coming to the passive artificial, develops the following: the infuse any kind of ready-made the immunoglobulins. For example, in case of rabies or any hepatitis, you are injecting immunoglobulins in order to prevent the infection to set immediately, because that active immunity takes some time. No, so meanwhile something should be protected. That's why we are inducing this kind of immunity. So here the two terminologies most important: premonition and concomitant immunity. That is the most important. Premonition means immunity may last, okay, as long as the microbes is present. For example, if the person has the if the person has infected with the syphilis or plasmodium, so the infection, once it should be completely cured, then only the syphilis or malaria infection will reinfect. There is no cure of the disease. The reinfection will not take place. This is called premonition. The immunity, this kind of the immunity called premonition. Okay. Second thing, even though there is an infection, for example, Haemophilus ducrei is there, the reinfections of the Haemophilus ducrei is possible. Okay. So we have a two kind of immunity: premonition and concomitant immunity. Once the infection is there, reinfection can be happen. That is called that is happen in the Haemophilus ducrei. Once the infection is there, the reinfection is less chance. Okay, that is in case of syphilis and malaria. And coming to this role of the passive immunity, in case of immunodeficiency disorder, because the patient, that immunodeficient person not able to that immune cells or immune mechanism cannot be takes place in the host. So in that condition, you can able to insert the, or you can inject the passive immunity. In case of post exposure, which I already mentioned, that rabies or hepatitis, you can able to only give the ready-made immunoglobulins, and passive immunity develops faster. That is because there is no lag phase or negative phase. What is meant by lag phase, we will discuss later. There is no immunological memory, so it is not useful for the, it won't develop the immunity again. The booster dose are not effective. Okay, let me discuss difference between the active and passive immunity. Active immunity produced by the host itself. Takes example, I got in the chicken pox, my immune cells get activated, I have developed immunity. It is because of my immune cells. In case of passive immunity, the immunoglobulin plays a major role, and my immune cells not participate in that. So it is the host immune cells are not participating. The induced, that is induced by infection or vaccination. It is induced by mother to fetus, that is passive and naturally, and ready-made antibody, passive artificially. It is long lasting in case of active immunity because my immune system get activated and it is produce the immunity. There is a lag phase because of the once the antigen are exposed, my immune system get activated. So it takes some time to produce the immune cells. It has a memory also. It needs booster. It has the negative phase also, and it is less chance of that immune deficient individual. It is not useful. In whereas in case of passive immunity, there is a last only shorter period. Okay, that active immunity will take some time more. Meanwhile, this passive immunity will play a role. Because of this only, there is no lag phase because this immunoglobulins won't able to act in the immune system. So there is no memory. There is less chance of getting the booster dose. So this is the primary and secondary immune response. Here, this is the phase called a lag phase. So once the antigen are exposed, the antigen gets stimulated by the antigen starts stimulating the immune cells, the T or B cells. Okay, so it takes some time to get activated. Once it is activated, B cells start producing the B cell get produce the plasma cells called IgM. This is called log phase, LOG, log phase. Okay, so it will rises. One point of time there is a stato phase. Okay, there is a steady state. Okay, so here the antibody production will be stopped, and once the infection is stopped, once infection are killed by this kind of antibody, there is a decline in the antibody protection. So this is called a primary immune response. First time the one person or patients are got infected with the antigen. So first started with the lag phase and the log phase and steady period, then decline period. The same antigen, it encountered the same person, it is called a second exposure. It means there is some amount of minimal amount of antibody will be left over. So once the antigen get exposed, this antibody, whatever the leftover antibody will react to that antigen. So meanwhile, the memory cell which is present that get activated so that the antibody titer goes up. This time there is a increased level of IgG. In here, the first time it is got exposed, so IgM has been raised. But here there is a chance of activating by the memory cell. So it has some memory. So straight away it produces the IgG antibody. This is called a secondary immune response. So this is the difference between the primary and secondary. Okay, the immunity against the first time, if the patient exposure to the antigen called a primary. If the second time, that is called a secondary. It will be slow or sluggish and lag period will be longer because first time it get activated. Whereas here, the lag period will be absent also or shorter because already it got activated. No negative phase because there is no antibody against that kind of antigen. Once it is get activated, there will be minimal antibody so that there is a negative phase. So there is a slow titer, initially IgM type. There is a high titer and IgG type. And here the B cell initially will be naive B cell. There is no activation at all. So once it got activated as a memory cells. So if the person has encountered second time, it will have the memory cells. Okay, this is the difference between the primary and secondary. What are the cells which they will help to bridge the innate as well as acquired immunity? So here there is some cells like a macrophage, dendritic cell, complement. These are the cells, even though it is a innate cells, it will participate, it grabs the antigen and present it to the acquired cells. That is BRC cell, T cells. What is the combined immunization? Let's take an example. For example, if the patient got infected with the rabies, they immediately we are giving both active as well as passive. So in order to prevent the entry of infection, we are giving passive immunity at the site. And also we are giving the immunity against the further development. That is called the active immunity. So in one side, we are giving active immunity as per the schedule. In other hand, we are giving the passive immunity, that is ready-made antibodies. And what is a local immunity? That is a some IgA secretory antibody which is present in the mucosal surface. Mucosa is a site the bacteria or any kind of things will enter and it will fight. So in that area, we have a secretory IgA antibody which helps to prevent the entry of the microbes. It is more useful in case of polio viruses. That's why we are giving polio, oral polio, so that the intestinal polio will be get eliminated. What is meant by herd immunity? Herd immunity means overall immunity of the community. If suppose we are immunizing the 60% of the population, we are expecting, because of this herd immunity, we are expecting the 100% of the immunity in the community. Okay, so this is the called herd immunity. So herd immunity develops effective vaccination has achieved or not. We are checking that. So if suppose 60% we are in the given immunization, the rest of the 40% got immunized because of this 60%. So for example, the polio vaccines we are giving for almost 60%, because this polio vaccine got excreted through the fecal. So this fecal excretion, because of the virus has a tendency to spread fecal orally. So other person will get the infected. The infected virus become vaccinated virus, so that they got vaccine. Okay. So what do you mean by some kind of specialized cells will helps in the adaptive immunity also. Okay. Let me discuss the questions. Okay, which is not mediated the immune immunity. We know already there is a T cells, right? Primary immune response is correct statement. Primary immunity, already I told you, it is a slow, sluggish, no lactate, antibody producing cells, uh, memory cells. What is the correct statement they are asking? So slow and sluggish only, because lag period will be there and IgM type. Okay. Toll-like receptor, it's a component of, you know already, it is a innate immunity. Coming to the acute phase reactant protein are all except, you know already what is that? Yes, coagulation. So in summary, we have discussed the immunity definition and difference between acquired and adaptive immunity. What are the cells are involved in the innate immunity? What are the cells are involved in the acquired immunity? And what is the primary immune response and secondary immune response? Okay. So these are the topic today. Kindly read and prepare for the short notes. Thank you.